Hi guys, welcome back to our channel.
In this video we'll be talking about cloning.
We're talking about what is cloning, why we use cloning, the different types and how it's used. Hope you enjoy!
What is cloning? Cloning is the process by which an exact, identical copy of the original can be made. Clones are those organisms that have identical genes. That means the sequence of their DNA is the same. Cloning is used in two contexts, cloning a gene or cloning an organism. Cloning a gene means to extract a gene from one organism and to insert it into the second organism where it can be used and studied. Cloning a gene sometimes can refer to success in identifying a gene associated with some phenotype. Cloning an organism means to create a new organism with the same genetic information as an existing one. This can be done by somatic cell nuclear transfer, in which the nucleus is removed from an egg cell and replaced with a nucleus extracted from a cell of the organism to be cloned. Why is cloning used? Cloning can be used in experiments aimed at understanding disease and developing new treatments for disease. The main reason that the scientists at Roslin wanted to be able to clone sheep and large animals was connected with their research aimed at producing medicines in the milk of such animals. This technique has been successfully performed on several species, such as frogs, mice, sheep and cattle. Different types of cloning. There are three different types of artificial cloning: gene cloning, reproductive cloning, and therapeutic cloning. Gene cloning. Gene cloning produces copies of genes or segments of DNA. Researchers use cloning techniques to make copies of genes that they wish to study. The methods consist of inserting a gene from one organism, often referred to as foreign DNA, into the genetic material of a carrier called a vector. Examples of vector include bacteria, yeast cells, and viruses, which are small DNA circles carried by bacteria. After the gene is inserted, the vector is placed in lab conditions that prompt it to multiply, resulting in the gene being copied many times over. Reproductive cloning. Reproductive cloning produces copies of whole animals. Dolly the sheep is an example of this, which will be further explained later in the video. Therapeutic cloning. Therapeutic cloning produces stem cells for experiments aimed at creating tissues to replace injured or diseased tissue. Therapeutic cloning involves creating a cloned embryo for the sole purpose of producing embryonic stem cells with the same DNA as the donor cell. These stem cells can be used in experiments aimed at understanding disease and developing new treatments for disease. To date, there is no evidence that human embryos have been produced for therapeutic cloning.
Plant cloning. Clones are groups of plants that are identical to their one parent and that can be propagated asexually, which is multiplication without passage through the seed cycle. It is also the best way to maintain some species. The major methods of asexual propagation are cutting, layering, budding, and grafting. Cutting involves rooting a piece of the parent plant. Layering involves rooting a piece of the parent and then cutting it. Budding and grafting is joining two plant parts from different variations. Human cloning. After the success of Dolly the sheep, the first animal to be cloned, why aren't we cloning humans? It's due to scientific, ethical, and commercial reasons. I think no one realises how hard cloning would be in some species, as it's relatively easy in others, said legal scholar Hank Greenlee of Stanford University. Cats easy, dogs hard, mice easy, rats hard, and humans are very hard. There has also been no commercial motivation for human cloning. Both the assisted reproduction IVF and other industries immediately said they had no interest in human cloning.
How cloning is done. Reproductive cloning is the process by which a whole organism is cloned, for example, Dolly the sheep. First, a cell is taken from the organism that is being cloned. The DNA from this donor cell is then transferred to an egg cell whose DNA has been removed. The egg cell is activated and begins dividing as if it were fertilized. An embryo results, and this embryo is then transferred to the uterus of a surrogate female. After gestation is complete, the surrogate will give birth to the clone, which is a genetic copy of the animal from which the original cell was taken. The first animal to be successfully cloned was a sheep named Dolly, who was born in 1996. So far, cattle, chickens, dogs, cats, horses, and several other mammals have been cloned.
Hi guys, hope you enjoyed this video. We had a lot of fun making it.
Make sure you catch us next time on our bio adventures.
And don't forget to like and subscribe down below.
Thanks, bye.
I said subscribe wrong again. Doesn't matter.